So we're going to take a look at nucleotides. Okay. Now, way back when we talked about nucleotides, and you've probably also learned the terminology nucleoside. This is way back when, like Biology 109. Do you know what the difference between a nucleotide and a nucleoside is? Yes, these are way back when. Okay. There are 3 things that make a nucleotide, and there are 2 things that make a nucleoside. Okay. One for both of them is a base pair. Okay. The other is a sugar, and it's ribose or deoxyribose. The last one is whether it has a phosphate or not. The nucleotides are the ones that have phosphates, and the nucleosides are the ones that do not. We're going to see nucleotides and nucleosides here. What are the 2 nucleotide base pairs that we use? The big classes. Pyrimidines and purines. Okay, so we're going to look at those syntheses of those 2. How many nucleotide base pairs are there in normal nucleotides? How many base pairs do we have to know? 4? 5. Yes. Ah. Okay, 4 in DNA, 4 in RNA, but 1 is differentiated between the difference. The 3 are the same. Why in the world do we look at nucleotides? Well, obviously genetic material, that's what we think about first. But they're also activating intermediates, meaning that they pose a lot of derivatives for activated intermediates. They're energy, mainly ATP, but we've seen GTP, we've seen CTP power things, and we are going to look in and see that UTP can power things. They're also components of just about everything we use. So when we see NAD+, or when we see FAD, or when we say CoA, we use those acronyms, but nucleotides are a portion of that active molecule, and they are regulatory, okay? Like cyclic AMP is a second messenger. If I say second messenger, what am I talking about? Where's the classification if I talk about second messenger? Oh, these are things that me and Dr. West love to teach. Signal transduction pathways, okay? And so they are produced as a part of a response, then goes on and stimulates other entities within the cell. Okay, so they're everywhere. And so what we're going to do is we're going to synthesize those bases. But what I want to introduce to you is a molecule called PRPP that is important in both of those synthetic pathways. PRPP is 5-ribosyl-1 pyrophosphate. Okay. Where does the ring structure come from? The ribose. Where do we get it in our synthetic pathways? Where do we get ribose from? The pentose phosphate pathway. And where do you derive the starting materials for pentose phosphate pathway? What did we start off with? I'm going to give you that assignment again. We started with a number of what molecules? What? I feel like I have done such a great job. We started with glucose. Not confidently, not with, you were throwing it out there, okay? Yes, you need to be confident. You know, you got 50-50 chance, you can be right or wrong, right? Okay, so we start off with glucose, okay, or glycotic intermediates or gluconeogenic intermediates, but that is also from glycolysis, and we can make the ribose. And then phosphates come from ATP, and we're going to create ribosyl-1 pyrophosphate, okay? And in this particular reaction, where are we going to attack the ATP? Will we attack at the alpha, at the beta, or the gamma phosphate? Okay, well, say it with confidence. Beta, yes. This is very rare for us. Okay? We will take the proton off the hydroxide. We will attack at the beta. Now, we know this because we transfer 2 phosphates, and what leaves is AMP. And that's just not an attack that we see very often, although it does happen. All right. And so, big, let's see, do you remember? If something is below the ring or above the ring, there's a designate for that, either alpha or beta. Where are these phosphates? Are they alpha or are they beta to the ring? They are alpha to the ring. Very good. All right. Now, when you do purine synthesis, this is probably, wait a minute. Okay, what does that mean? Okay, where we're going to get the individual atoms for this ring system. And if you take a look at it, there's one thing that you can recognize. Almost all of the atoms in the ring are coming from single or one at a time additions. There's only one thing that is a base, and that's glycine, that only brings in 3 of those atoms. All right. Secondly, this is one way it can be written. Unfortunately, on standardized tests, some of these things can be written different ways, like this molecule. What is that? CO2, right? What can it be found in the body as? Bicarbonate ion. And so some text will use the bicarbonate ion, some will use CO2. They're interconvertible, so don't dismiss that. Okay, when you look at it, it says, oh, there's no answer. Well, it was just represented differently, and we've seen that represented differently a lot of different places. Okay, and so we've got lots of different pieces of the puzzle. All right, the first thing that we're going to do is a transferase, and we're going to have a reaction in which we have glutamine going to glutamate. This is going to be seen a lot. So we start off with glutamine, we go to glutamate, and we have water. Okay? Effectively, the water and the glutamate react first, and what we're going to get is an amine coming off, or basically ammonia attacking. Okay? And so this is a way in which this ammonia is being carried by the glutamine to be used. Okay? And this is the ammonia that is on the R group. Okay? If I take this off and make a acid, I have glutamate. So we're going to see this a lot. Now this amine then can be used to attack. I'm going to show that second reaction with different arrows. Okay. What kind of reaction did I draw using the red arrows? E1, E2, E2 prime, SN1, SN2. Got a two fingers. Okay, so we're narrowed down to E1, E2, E2 prime, and SN2. SN2. Give me another rationale without looking at the arrows, just looking at the reactants and the products. Why would I say that it is an SN2 attack? The amine is in the beta conformation. It's inverted. The stereochemistry is inverted from the beginning to the end, where the nucleophile attacks. Okay, the leaving group it's an opposite. Now, one thing about nucleotides, all the nucleotide base pairs are in the beta conformation, and so this is your first molecule, or the first atom within that ring structure. You can see that double ring right there. Okay. Okay. All right. Name for me this molecule right here. What is this? It's a bunch of lines. It looks like what? Glycine. Very good. All right, glycine. That's a good thing to know because I expect you to be able to recognize every amino acid. I actually even expect you to be able to draw every amino acid. Okay? And so glycine plus ATP is the first reaction. When glycine reacts with ATP, will it react at the alpha, the beta, or the gamma? Gamma. Okay? Now, red, yellow, blue. Which is the gamma, red, yellow, or blue? Red. Okay. What have I done to the glycine at this point? What do we talk about when we covalently modify a molecule this way? We've activated it, we've transferred the energy from ATP into that intermediate, and essentially we've made a good leaving group. So the glycine is now activated. All right, looking at this reaction here between the ring structure and the glycine. Here's the product. What do you see different in this ring structure? Any difference here? There? Here? Anything around on the ring? This hydroxide, that hydroxide? This nitrogen? Yes. What is different between this nitrogen and that nitrogen? It's bound to a carbon, right, and lost a hydrogen. All right, where is this ketone? That ketone, right? So I have to be attached this to that, and to do that, I've got to remove this. Okay, negative charge, it's a nucleophile. It attacks at the carbonyl because that is a wonderful electrophile. Kick up, kick down. I have a wonderful leaving group. And now I have 4 atoms within the ring structure. All right. So this is the largest addition of atoms that are part of the ring structure. Now what is interesting is that we have this right here, which is not in the ring structure, so eventually that's going to have to go away, but this nitrogen, that carbon, and that nitrogen is part of the ring structure. All right, THF. Remember, THF is a very large molecule. Before, we had a methyl group attached to this nitrogen called the 5th nitrogen, and it was a methyl carrier. Okay. Here we're going to have a formal group, which is also a 1-carbon intermediate that's attached to the 10th nitrogen. And so this is what's going to be transferred, and it's going to be transferred onto, well, where is it transferred onto? In the product? Onto a nitrogen, and you can see that this nitrogen here is that one. It has 1 hydrogen, here it has 2, and it's attached to the ketone. All right. Red or blue? What? What was that answer? Yes. Either. Why is it either? They're equivalent. Okay? All right, good. Attack. Kick up. Kick down. And this will pick up a proton. All right. I am getting closer to a ring structure. Okay, one of the rings has 6 atoms as part of the ring, and let's take a look. 1, 2, 3, 4, 5, almost there. Okay, 5 atoms, 1 bond, 2 bond, 3 bond, 4 bond, 5 bond. So we're getting close. We also have a 5-membered ring in the structure. All right. I told you that I react glutamine and water, and I get glutamate and an amine coming off. That was that first reaction. Okay. This is a little complex in its reaction. Okay. Here is the reactive species right here, but I also have ATP right there. All right. It's going to create a really kind of strange intermediate, NH3. If by chance I take an amine off, okay, I am going to get an attack right here. Okay, I'm going to attack a carbonyl. Tell me, what can I do when I attack a carbonyl? What must I do? Kick up. I could then what? Kick down, or I could kick up and pick up a proton. The other thing that can happen is it can kick up and attack the ATP at the gamma phosphate and become phosphorylated. So I'm going to change the slide. I'm going to write the carbon I just attacked. I'm going to put the nitrogen I just put on there. I'm going to put the oxygen that was the carbonyl now attached to a phosphate. Okay. And this carbon has an R group on the side, that's the ring, and an R group on the side, that's the ring. Okay. So this is the intermediate structure of what I'm looking at. And if I take a look, again, this is where we're heading. You can see that in my intermediate I have a nitrogen with 2 hydrogens, but in the product I have a nitrogen with 1 hydrogen. So I have to get rid of one of them. I will form the double bond between the nitrogen and the carbon, which is this one right here. It's a very strange reaction. Okay, this would be something more than I would ask you on the exam. Alrighty. What in the world is happening now? All right. We are going to close this ring. This is the 5-membered ring. All right. What in the world has changed? What have we lost? Right around here, what have we lost? That proton's gone. Anything change? Anything change? Anything change? On that nitrogen? No. It's got nitrogen, 2 carbons, and a hydrogen. Nitrogen, 2 carbons, and a hydrogen. What about change here? Oh, yes. That's a change. This nitrogen that had a hydrogen is now connected to a carbon that becomes a hydroxide. That's the one that comes up the hydroxide. Okay. I've closed the ring. That's all well and good. But there are going to be 2 reactions that occur. Okay. The ring is closed, but I can make this just a tad more stable. Okay. If I place a base there, okay, what do we see in this ring that is different than that ring? I got right here a double bond, right? And this nitrogen is missing a hydrogen. This carbon is missing that hydroxide. And what leaves? Water. Do I have water written? I should have written this. Okay, that's great. Now this last reaction, okay, occurs, and what is happening in this last reaction? All right, so this seems to be fine. That nitrogen seems to be fine. This is still a single bond, right? But we get some changes right in here, right? This carbon has how many hydrogens? 1. How many here? 2. Okay, so obviously I'm going to do something there. And so if I have too many, I'm going to take and deprotonate it. I will then form the double bond. But what's wrong with this carbon if I form a double bond? 5 bonds. And so what am I going to do with that pair of electrons? Grab a proton. Now, this step here, or that ring structure, has some special stability. What is the specialized stability in that ring structure? Resonance. What kind of specialized resonance? Aromatic. Okay. It is aromatic because of those two pair of electrons there. That looks like one little thing on the slide. Okay, 2 pairs of electrons. So we have an aromatic ring. All right. If you go back to that first slide, it said CO2 is the carbon that comes in. But look at the reactant. It's what? Bicarbonate. And so even in the mechanism, they actually start with bicarbonate and it reacts with ATP to create an intermediate structure that is the activated. All right, and so what's going to happen? Where in the world do I add this? Well, this CO2 right here, where this phosphate is the leaving group, is going to be added right to that nitrogen. And we've added the acid. 1 carbon added. Okay. This is a real complicated mechanism because do you see what happens? The CO2 moves. It shifts from this nitrogen. It even bypasses this carbon and goes way up there. There are cofactors, there are multiple steps, and so this is not something that I would ask you to know in terms of the mechanistic steps. But this one I would. All right. Tell me, before we do the mechanism, what kind of enzyme, what class of enzyme takes these 2 reactants and produces this product? A kinase. Okay. And I will attack at the gamma phosphate. To make an activated intermediate. Now, I'm going to add aspartate to this. And if you go back to that slide, the aspartate is on the slide and it says it's only going to donate the nitrogen. So we're going to add this entire big molecule on there, and what's eventually going to just be added is 1 nitrogen. And so if I look at the reactant up here, I've just activated this. That means I'm probably going to attack this carbonyl. And I look down where that carbonyl is, and now it's not attached to a phosphate anymore. It's attached to a nitrogen of the aspartate. 1 proton, 2 protons. Take off a proton. Attack. Up, down, and out. Now that looks like a really big molecule on there that's stuck onto our growing ring structure. Okay. All righty. What in the world is going to happen here? Fumarate falls off. All right. And we're left with 1 nitrogen on the growing ring structure. All right, if you do some inventory, here in this there are 2 protons. Most of the time it won't even be written, but you would need to put the protons on there. You can see here the acid carbon 2 protons, the acid carbon 1 proton. How many protons are here? At that 1, still 1, and then an acid. Got to take that one off. It forms the double bond, and that picks up a... What kind of reaction is this one? E1, E2, E2 prime, SN1, SN2. E2. Okay, fumarate. What pathway are we in with this? Citric acid cycle. Do you know one other pathway that we're in? Urea cycle also, in the bicycle of the urea. All right. We are continuing to build the ring one by one by one. Tetrahydrofolate will come in very similar to the last reaction where we're transferring a formal group. And again, we're going to transfer it to a nitrogen. Okay, here's a nitrogen with 2 hydrogen. Here's a nitrogen with 2 hydrogens. We look at the product. Here is it sitting on the bottom one. Whoa, well that was weird. Got a little free with my arrows. Okay. Why do I add that formal group to the bottom nitrogen and not to the top nitrogen? In actuality, the top nitrogen may be a little bit more reactive because the first step is deprotonate, right? The nitrogen that is attached to the carbonyl is actually more acidic, easier to deprotonate than the one that is down because there's a resonance form. Ah, THF is kind of big. Yeah, that is an important aspect, but both of them are primary amines, so they should be able to have similar access to primary amines. Why in the world do I only attach there? What is it?
The enzyme.
Yes, it's all magically the enzyme. Remember, these reactions are oriented. What would you predict if you were in an organic flask? You would show both. And you may even say 50-50, but the top one actually is more acidic, so you would probably get more reaction at the top than the bottom. And so the enzyme is controlling why we have 100% reaction to only one product when there is 2. And we never forget this when we're talking about biochemistry. Or we shouldn't. Let's put it that way. We shouldn't forget this. All righty, we're going to close the ring again. Okay. In this ring closure, we're going to again attack an amine and a ketone. So the amine and the aldehyde. And so we are going to attack this amine, which is this one, and this ketone, which becomes this hydroxide. This amine only has 1 proton, so let's take it off. Attack. And pick up a proton. Why did I pick up a proton? Because I created a hydroxide. I could have kicked down, but I didn't see a ketone in the product. I have closed the ring, but there's a way to make it more stable. And to do that, if I take a look at the product final and the reactant, what am I missing here? Between there and there? An H and an OH, which makes dihydrogen monoxide. You're right. Only a GChem student would know that. I have a nucleotide. Have you ever seen IMP? Okay. It is not one of the standard 5 in either DNA or RNA that we think of, but in actuality, in RNA, there are some derivatives of our main base pairs, and sometimes you will see I there. Okay, it's also part of a breakdown product, it's part of the synthetic product. And so I have a purine. I just don't have one of the two that I normally think about. Okay. The inositate is a branch point. Okay. And it is IMP because it has a ribose and one phosphate. All right. But we are going to create AMP and GMP from this. All right. We're going to branch and we're going to start synthesizing AMP. Now, what's interesting about the synthesis of AMP, the first powering molecule for AMP synthesis is GTP. To make AMP, you have to have GTP. Now, I write this a little bit differently. I write GDP and then the reactive phosphate. Okay. And where in the world is that thing supposed to go? It's supposed to go onto this oxygen, which is that oxygen. Okay. Well, what in the world? Okay. If I've got to get this double bond broken, okay, then what's different? Well, it's not any different here, but it is different there. That's a single bond, that's a double bond, right? Can anybody tell me how in the world, I still have a proton, how do I make a double bond from this to that? Lone pairs. Something that is there that we don't write but that we know is there can form the double bond, allow the electron pairs to go onto the oxygen and then become a nucleophile and attack. And that may be something that you might get lost because it wasn't articulated, but you need to be able to write lone pairs where they are. You need to be able to write the protons where they are understood to be, because sometimes you might find there are more protons on one side than the other. So being able to do that's important. Okay, we've got another aspartate come in. All right, and we're going to react it. Now, this is kind of a really strange reaction. I know where that thing's supposed to end up, right? It's supposed to be on this carbon. Got it? That's that carbon, right? And so I also have a nitrogen that has two hydrogens. I have a nitrogen that has one hydrogen. So this is kind of weird. Watch this. I'm going to take that off. Okay. And I'm going to attack that carbon because that's where it needs to be connected to. And what happens is the electrons kick up onto the nitrogen, making it neutral. But I don't see them neutral here, okay, in the product. What then happens, okay, is this. Look in the product. What am I missing? A proton. So I'm going to have, at this point in time, a base, take that off. I will reform the double bond, and the phosphate leaves. Adenyl succinate. All right. Are we ready? In this case, succinate will come off. And to do that, very similar to the reaction that we had with aspartate before, I take that proton, it creates a double bond, and I pick up a proton, and I now have AMP. All right. Back to an acetate, the IMP. We're now going to start making GMP. All right. In the reaction of to create AMP, we needed GTP as the powering molecule. So watch the synthesis as we go. We're going to encounter something. Now, the enzyme within this, the synthesis of GMP has a reactive sulfur, and it will be covalently part of the reaction. So the sulfur is going to attack, and we can obviously see it's going to attack that carbon. And so I'm going to attack it at that carbon. What happens to the double bond? It changes around the ring. But if I change that one, what has to happen? Changes the other one. And if I do that, then what has to happen? This has to pick up a proton. Okay. That's fine. All right. What is the normal name a class of enzymes that converts this reactant into this reactant that uses this cofactor? A generalized enzyme name. Dehydrogenase. Okay. And this is an oxidative reaction. And where will I start? Take the proton off, deprotonate. So I'm going to take that proton. Where does that pair of electrons go? Double bond. Well, what does that do? Moves this double bond in the ring. Well, what does that do? Moves this back here. And what does that do? A hydride leaves. If you say a proton leaves, that would be wrong, because I have to also have the electrons go. Okay. I've now connected the reactant to the enzyme and placed the sulfur as part of that. I've also had an oxidation event occur. All right, what in the world is happening now? I'm going to add water. Okay. And so the water goes onto the carbon that has the what? That sulfur, the cysteine. So I'll take this. I will attack it. Okay, but what happens with that? Moves the double bond over, which then does what? Moves the double bond over, which then does what? Picks up a proton. Now, moving those double bonds around and picking up a proton, what I've done is I've allowed it to attack, and there's no charged species. It's all neutral. So that's pretty good in terms of reducing the energy required for this reaction. All right, that's not exactly where I want to be, right? And so what I'm going to do is, in this case, take the enzyme and make sure it goes away. So to do that, what do I have to do? Take the proton, double bond, move it on the ring, move it on the ring, and what falls off? The sulfur of the cysteine in the enzyme falls off. And now I am in an entity that can form a resonance structure, okay? And we will do that resonance structure by taking this proton, filling a ketone, creating a ketone, and that picks up a proton. And I get to something called xanthine. Right, I'm trying to get to GMP, not there yet. Okay, I'm waiting, making sure. We good to go? Okay. GMP in 4 easy steps. All right, here. To get to GMP, I have to use an energy molecule, and that energy molecule is ATP. To make AMP, I have to have GTP. To make GTP, I need ATP. They are reciprocally regulated. So they're balanced between each other. All right, where in the world am I going to attach this? Well, I've got to get it to this oxygen. That's oxygen. Okay. What is different? There's a double bond. Not a double bond. Make a double bond. Okay. This will then attack, kick up, kick down, and ADP is the leaving group. Again, you should recognize glutamine, glutamate, add water. I'm going to have this amine as the leaving group. So this is a way to carry an amine over and make it reactive. And the reactive is attack this. Okay. No. Ah. Oh, it did. It'll kick up on the nitrogen. It will behave like a ketone, and the phosphate will leave. And I have GMP. All right. I have made AMP and GMP, 2 of the 5. Are these RNA or DNA? Are these components of RNA or DNA? They're not both. You find G and A in RNA and DNA, right? But what's the distinguishing factor between RNA and DNA? It is RNA because the sugar has both hydroxides. And so, just to realize. Okay? The pyrimidine synthesis is a little bit easier in that there's only 2 things. Aspartate itself, so the amino acid, and carbamylyl phosphate. Carbamylyl phosphate was seen where? Which cycle? The urea cycle. Okay? And so it looks simpler. Things that look simpler, always simpler? Not always. All right. So carbamylyl phosphate and the aspartate will react. And that looks a little complicated, doesn't it? But it really isn't that complicated. All right? It's just twisted around a little bit. So when you do your inventory where you're attaching things, all right, this nitrogen to a ketone looks like this nitrogen to a ketone. And then it's not attached to a phosphate anymore. It must be attached to a nitrogen. And the only nitrogen here is there. Okay, what happens is this whole part of the molecule is now written below. This acid is this acid. It's probably the most complicated part of this slide. All right. If by chance you get through this entire semester in my portion, if you cannot allow a phosphate to leave off a ketone, I'm going to be very, very disappointed in you. Okay, if you draw that part wrong. How often have we drawn that attack? A lot, okay? And so please, take home, in your heart of hearts somewhere, put it, please. All right. Now, interestingly enough, we do react a ketone with an amine a lot of times because the ketone can create an intermediate that has a negative charge on an oxygen. It's okay. What we don't do is attack an amine to an acid very often because the acid is in resonance. And so by attacking and putting another charge, then you'll have 2 negative charges on 2 oxygens that are very close together. And so that kind of attack doesn't happen very often unless an enzyme extraordinarily stabilizes that. The cofactor for this enzyme has the zinc ions. And what happens is the zinc ions will coordinate to those negative charges in the intermediate such that it isn't a resonating structure as you think about an acid, and those positive charges will allow this reaction to occur. So we can force something that wouldn't happen necessarily in an organic flask happen inside an enzyme because of these specialized stabilizing events. And so what we're going to do, we're going to show the stabilizing event where those negative charges are actually coordinated to the metal centers. Now, in this case, you're going to see a hydroxide that can leave as part of this reaction, and what actually does that is weird. I'm missing something. I am definitely missing something. Okay, I'm going to go back and look at this slide. I think I might have written something wrong because, yeah, it's there, but why would it leave? Why would you break a bond? Ah, no, I'm not missing something. I figured it out. Sorry. This is what happens. That pair of electrons falls back into the form of the double bond, and then this picks up a proton. We usually don't have a hydroxide leaving, but what happens is that hydroxide is still coordinated onto that metal center. Eventually, that hydroxide, once it leaves the active site, will be converted into a water. Okay. Ooh, interesting, something that we've seen. Let me go back one slide, and then I'll go back to this slide too. All right. The first reaction here is starting to build the base. If you go all the way back to the beginning of our synthesis for GMP and AMP, where did we start off with? What was the first structure I introduced to you? No, way before IMP. I went all the way back to ribose. In the synthesis of AMP and GMP, we start with the sugar and build the ring around it. Here, in the pyrimidine synthesis, we're going to build the ring structure and then attach it to the sugar. So this is a compare-contrast between the two. Okay. All righty. Last slide. It is a dehydrogenase reaction. We have an electron carrier, and we see here we have a single bond, we have a double bond. I have too many hydrogens. All right. Last question that will be answered when you see me next. Red versus blue. This is for you to think about. Which is right, red versus blue. I am in the middle of grading your quizzes. They are coming out very well. There's one or two little things on some of them, but I'm in the middle, so I'm working through. There's been a couple of errors. They haven't converted in the grading scheme on a couple of your a couple of your submissions, so I'm going to have to download it, convert it, upload it, but it's going to take me a little while. But I am working on them, and hopefully we'll have them done for you. But how did you like that quiz? A little bit different. I wanted to be a little bit more integrative because, yeah, you learn these pathways, but at some point in time, you're going to figure out how they actually really work, and these are real-life situations in which these apply to how this biochemistry actually works in the body. And so I wanted you to think through some of those. Some of them are a little bit difficult to think about, but it's a better way to think about this material. All right?